The webinar will begin soon. Please stand by. Good afternoon, everyone. My name is Laura Morell, and I work in the National Center for Emerging and Zoonotic Infectious Diseases at the Centers for Disease Control and Prevention. On behalf of CDC's One Health Office, I'm pleased to welcome you to the monthly Zoonoses and One Health Updates call on March 1st, 2023. Next slide, please. Although the content of this webinar is directed to veterinarians, physicians, epidemiologists, and related public health professionals in federal, state, and local positions, CDC has no control over who participates. Therefore, please exercise discretion on sensitive content and material, as confidentiality cannot be guaranteed. Today's webinar is being recorded. If you have any objections, you may disconnect now. Links to resources from each presentation are available on our website at cdc.gov/onehealth/zohu/2023/march.html. Next slide. Today's presentations will address one or more of the following 5 objectives: identify an implication for human, animal, and environmental health, identify a One Health approach strategy for prevention of public health threats, identify a One Health approach strategy for detection of public health threats, identify a One Health approach strategy for responding to public health threats, and list 2 ways to improve collaborative practice across the public healthcare team. Next slide. In compliance with continuing education requirements, all presenters must disclose any financial or other associations with the manufacturers of commercial products, suppliers of commercial services, or commercial supporters, as well as any use of unlabeled products or products under investigational use. CDC, our planners, presenters, and their spouses or partners wish to disclose they have no financial interests or other relationships with the manufacturers of commercial products, suppliers of commercial services, or commercial supporters. The Planning Committee reviewed content to ensure there is no bias. The presentations will not include any discussion of the unlabeled use of a product or a product under investigational use. CDC did not accept commercial support for this activity. Next slide. Instructions for receiving free continuing education are available at cdc.gov/onehealth/zohu/continuing education. The course access code is ZOHU webcast. To receive free CE for today's webcast, complete the evaluation at cdc.gov/tceonline by April 3rd, 2023. A captioned video of today's webinar will be posted at cdc.gov/onehealth/zohu/2023/march.html within 30 days. To receive free CE for the web on demand video of today's webinar, complete the evaluation at cdc.gov/tceonline by April 4th, 2025. Next slide. Before we begin today's presentations, Casey Barton-Baravesh, Director of the One Health Office, will share some news and updates. You can begin when you're ready.
Hi, greetings, everyone. It's great to be back for the March ZOHU call. We appreciate you all sharing the ZOHU call website link with your colleagues from public health, agriculture, wildlife, plant and environment, and other relevant sectors, and let them know about the live webinars, the video recordings, and the free continuing education that the ZOHU call offers. We would love to have your colleagues join us for these calls. So before today's presentations begin, I'll take a moment to share some updates and resources. And as always, you can find links to all of these resources in today's ZOHU Call email newsletter. And if you're not yet subscribed to the newsletter, all you have to do is go to the link at the top of the main ZOHU page and sign up there. You'll be able to find on CDC's website the latest COVID-19 guidance and resources for keeping people as well as animals safe and healthy. If you'll go to the next slide, please. Today's newsletter highlights a couple publications, including a recent Medscape article, One Health, a crucial collaborative tool for early disease surveillance that I authored, and also an emerging infectious diseases publication on the environmental persistence of Mpox virus on surfaces in the households of people with travel-associated infection in Dallas. Next slide. We've also shared links to web resources and announcements, including the National Nature Assessment website, a map of state laws on the sale of unpasteurized cow's milk from 2012 to 2019, and a map of antimicrobial resistance investments. Next slide. Other resources include a new workshop summary on prioritizing zoonotic diseases for One Health collaboration in Colombia. And next slide, there's also a statement on avian influenza in mammals from WOA, or the World Organization for Animal Health. We encourage you to check that out. And from APHIS, questions and answers on SARS-CoV-2 surveillance in free-ranging deer and other cervids is available. Next slide. Some events and observances of interest for March include a World Wildlife Day webinar happening March 3rd, hosted by WOA. March 5th through the 11th is Groundwater Awareness Week, and March 22nd is World Water Day. Next slide. Finally, in addition to the ongoing listeria outbreaks linked to deli meat and cheese and enoki mushrooms, there's a new listeria outbreak, and the food source for that one is still under investigation, but there's information available online. Next slide. You can visit CDC's Healthy Pets, Healthy People website for a selected list of ongoing and past U.S. outbreaks of zoonotic diseases. And you can also find details about ongoing U.S. outbreaks on the CDC websites linked from today's ZOHU newsletter. Our next ZOHU call will take place on April 5th, 2023. And please continue to send presenter and topic suggestions for future presentations to us. We look closely at all of those and work to get speakers lined up for those topics on our ZOHU calls. We're also happy to receive news from your organization, our key links. All you have to do is send that to us at zohucall@cdc.gov. That's ZOHUCALL@cdc.gov. Now I'll turn the call back over to Laura. Thanks again for being here today.
Thank you. Next slide, please. As a reminder, you can submit questions at any time using Zoom's Q&A feature. Please include the topic or presenter's name with your question. The Q&A session will follow the final presentation if time permits. You can also email questions to today's presenters. We've included their email addresses on this slide, on the ZOHU Call webpage for today's webinar, and in today's email newsletter. Next slide. So our first presentation, Sporotrichosis, Overview and Updates on an Emerging Zoonotic Disease Threat, is by Dr. Ian Hennessy. Please begin when you're ready.
Hey everyone, can you hear me okay?
Yes.
Okay, great. Well, thanks so much for having me today. My name is Ian Hennessy, and I'm an Epidemic Intelligence Service Officer with the Mycotic Diseases Branch at CDC. Today, I'll be providing an overview and giving some updates on an emerging zoonotic disease called sporotrichosis. Next slide, please. I'll start with a quick background about sporotrichosis. Sporotrichosis is a fungal disease caused by Sporotrix fungi. It affects numerous types of mammals, including humans and cats. Usually, infections are characterized by cutaneous and subcutaneous symptoms, including skin lesions, ulcers, and lymphadenopathy. In rarer cases, it can cause respiratory disease or disseminated disease, such as infection of the bone or nervous system. While the disease is rare and can usually be treated, it can result in hospitalizations and cause severe disease or death in immunocompromised individuals, such as those with advanced HIV disease. It is often more virulent in cats than in other mammals, which I'll discuss later in this presentation. Next slide, please. Sporotrichs fungi are widely dispersed in the environment, where they are found on dead and decaying plant material. The fungus has been isolated on hay, sphagnum moss, soil, decaying wood, and other plant materials. Sporotrichs are diamorphic fungi, meaning they undergo a morphological transition from a mold form in the environment to a parasitic yeast form in host tissue. Next slide, please. Sporotrichosis is an implantation mycosis, meaning it is acquired via traumatic inoculation. Rose gardeners, for example, can be infected after being pricked by rose thorns on which the fungus is present. For this reason, sporotrichosis was traditionally known as rose gardener's disease. Other outbreaks have been associated with handling hay and sphagnum moss. Forestry workers, tree nursery and garden center workers, and others who work with roses, hay, and sphagnum moss are considered risk groups for contracting sporotrichosis. Cats and other mammals can also be infected via contact with plant materials on which the fungus is present. Next slide. Cats can also transmit sporotrichosis directly to cats, humans, or other mammals. In the past, this transmission route was considered rare. However, cases of cat-transmitted sporotrichosis are increasing dramatically in Brazil and neighboring countries. This is primarily due to an emerging Sporotrich species called Sporotrich brasiliensis, or S. brasiliensis. This species was first detected in the 1980s in Brazil and is now causing the largest outbreak of animal sporotrichosis ever recorded. Next slide. In contrast to other types of Sporotrichs, S. brasiliensis spreads very easily between cats and from cats to other mammals, including humans and dogs. Cats can harbor the fungus on their claws, nail beds, and oral cavities, and it can be transmitted via scratches and bites, but also through non-traumatic routes, such as direct contact with the lesions from an infected cat, droplet exposure via sneezing, and potentially inhalation. These unique transmission modes increase the epizootic and epizoonotic potential of S. brasiliensis. Additionally, infected cats have very high fungal loads, making them extremely efficient at transmitting this fungus. Fighting, mating, and grooming behaviors facilitate transmission, and intact male free-roaming cats are especially at risk for acquiring and transmitting the disease. Finally, in contrast to other Sporotrich species, which generally infect mammals in the mold form, S. brasiliensis can be transmitted directly in the yeast form. This is a key factor in facilitating transmission directly between mammals. Next slide, please. Sporotrichosis caused by S. brasiliensis is more virulent in cats than other types of Sporotrichs. Cutaneous symptoms are the most common and include skin lesions, which are most often present on the face and in large lymph nodes. Respiratory symptoms like sneezing, dyspnea, and nasal discharge are also common and can be associated with more severe disease and treatment failure. Disseminated disease is less frequent, but generally has very poor outcomes in cats. Itraconazole is the first-line treatment and can be delivered along with potassium iodide. Treatment compliance can be challenging as treatments can last for months and therapeutic failure and recrudescence are common. Next slide, please. Zoonotic transmission of S. brasiliensis from cats to humans is common, with thousands of cases of cat-transmitted sporotrichosis reported in Brazil. Risk groups include people who have frequent contact with cats, such as veterinarians, animal caretakers, and cat owners. Clinical symptoms in humans often mirror what we see in cats, with cutaneous or lymphocutaneous lesions being the most common. Often these appear at the site of a cat scratch or other related trauma. Ocular involvement can occur when exudate from lesions from cats comes into contact with a person's eyes. Disseminated disease and central nervous system disease are more rare and often occur in immunocompromised individuals. Next slide, please. Cats can also transmit sporotrichosis to dogs. Hundreds of canine sporotrichosis cases have been reported in Brazil, the majority of whom had contact with a sick cat before their diagnosis. Infected dogs have lower fungal loads, though, so they typically do not pose a threat for zoonotic transmission to humans. Next slide, please. To give you a sense of the magnitude of this outbreak, this map shows feline sporotrichosis cases in Brazil in the 1980s. Next slide. Cases began to increase in Rio de Janeiro in the 1990s and spread to other parts of southern Brazil. The outbreak accelerated in the 2000s and reached northern Brazil by the 2010s. Next slide, please. By 2020, cases had been detected in almost every state in Brazil. Next slide. And the most recent map from 2022 gives you a sense of how explosively this disease has spread in cats in Brazil over the last 3 decades. A similar increase has been seen for cat-transmitted sporotrichosis cases in humans in Brazil. Next slide, please. Alarmingly, S. brasiliensis cases have increasingly been reported in countries beyond Brazil. Feline and human cases have been widely documented in Argentina, where S. brasiliensis is now also considered endemic. Travel-associated cases have been detected in Paraguay. Suspect cases have been reported from Panama and Colombia. And just this month, researchers reported the first cases of S. brasiliensis in cats in Chile. None of the cats had a travel link with Brazil, and 2 were stray cats, raising concerns that the disease may be more widespread in the country. Next slide, please. Last year, the United Kingdom reported its first 3 human cases, all of whom had been exposed to a cat who was brought from Brazil 3 years prior. These cases highlight the potential threat that S. brasiliensis poses for international spread via the importation of infected cats from endemic countries. The very long incubation period in this situation also highlights how difficult it can be to screen potentially infected cats at ports of entry. Next slide. In the United States, we have heard anecdotal reports of increased feline and cat-transmitted sporotrichosis cases in recent years. However, so far, S. brasiliensis has not been detected in the United States. There is a potential, though, for introductions via importation of sick cats from endemic countries. Establishment of this rapidly spreading zoonotic disease in the United States would constitute a serious health threat for domestic and community cat populations. Its establishment would also pose a health risk to veterinarians, animal shelter staff, cat owners, and others caring for cats. Travel-associated cases can also occur in individuals who travel to endemic areas. For example, a cat-transmitted sporotrichosis case caused by S. shankii was recently reported in a woman in Boston following contact with a sick cat in Brazil. Next slide, please. In the mycotic diseases branch at CDC, we've been working to understand the burden and epidemiology of sporotrichosis in the United States. We have also been providing technical support such as whole genome sequencing, antifungal susceptibility testing, and epidemiology support to partners in the United States and South America for sporotrichosis case investigations and outbreak response. We're also in the early stages of developing a prospective surveillance study to monitor Sporothrix prevalence in stray cats with the goal of better understanding the epidemiology of feline sporotrichosis and detecting S. brasiliensis if it is introduced in the United States. This year, we also helped establish a global sporotrichosis working group, which aims to increase communication and awareness of the evolving zoonotic sporotrichosis situation in South America. Finally, we are working to raise awareness about this emerging zoonotic disease through outreach to veterinary, clinical, and public health communities in the United States. Next slide. As we've seen, sporotrichosis has a complicated transmission cycle involving a widely dispersed environmental reservoir and epizootic and zoonotic transmission. A One Health approach is therefore needed for controlling cat-transmitted sporotrichosis, involving the contributions of multiple disciplines. Veterinarians and others involved in cat care have a critical role to play and can be instrumental in early detection of cases and risk communication to cat owners. Early treatment is also critical for reducing fungal burden in infected cats and risk of onward transmission. If local cases are detected, public health personnel can work with veterinarians to support outbreak response and educate clinicians about how to recognize and treat potential human cases. Collaboration with environmental scientists and microbiologists is essential for collecting, storing, shipping, and testing of environmental and clinical samples. And border health personnel can also screen cats from endemic areas for potential signs of sporotrichosis. Next slide, please. In conclusion, cat-transmitted sporotrichosis is spreading rapidly in Brazil, and cases are increasingly reported in other countries. There is a potential for international spread, including to the US, via importation of sick cats from endemic countries. A One Health approach is necessary for controlling this disease, which involves the collaboration of diverse disciplines. And lastly, I want to ask the veterinarians and clinicians on the call today to please notify their local health departments if they see suspect cases of cat-transmitted sporotrichosis or other unusual trends in sporotrichosis cases in their clinics. We would also love to hear about it at CDC, so please do reach out to us as well. Next slide. And with that, thank you so much for listening, and I really look forward to taking your questions at the end of the ZOHU call. Thank you.
Thank you. Next slide, please. And next slide. Our next presentation, Mpox response and considerations for animal testing, is by Jeffrey Doty. Please begin when you're ready.
All right, sounds great. Thanks, Laura. Hi, everybody. I'm Jeff Doty. I'm the Ecological Investigations Unit Lead for the Pox Virus and Rabies Branch, and I've also served as the One Health Team Lead for the CDC Mpox response. Today, I'll be giving a little bit of an overview about the Mpox response at CDC and One Health, as well as the considerations for testing animal samples. Next slide, please. So what is Mpox? I guess, first of all, Mpox, well, the virus is called monkeypox virus. However, recently, WHO has changed the name of the disease from monkeypox disease to Mpox. So throughout the course of this presentation, I'll just refer to both the disease and the virus as Mpox for simplification. So this is a member of the family Poxviridae in the genus Orthopoxvirus. It is a zoonotic virus, meaning it can infect both humans and animals. Formerly rare, this has mostly been spread in West and Central Africa prior to spreading wildly in the 2022 global outbreak. WHO did declare this a public health emergency of international concern in July of 2022, which still continues today due to some continuation of cases in certain countries, including countries that are historically endemic. Next slide. So this 2022 outbreak we're seeing right now, I mean, how is this different? Well, number one, this is the largest number of cases seen in countries outside of Africa. Many of the cases we're seeing now, almost all of them, have no obvious travel link to endemic countries in West or Central Africa. However, at the beginning of the outbreak here in the United States, many cases were associated with international travel, primarily to Europe, and then bringing the virus back to the United States. Since then, we've seen many cases domestically acquired here in our borders. There's also a significant correlation between men, and specifically men who identify as gay, bisexual, or other men who have sex with men. We don't think that this is necessarily related to the actual sex act itself, but the network of close contact that occurs within this population. Some symptoms have been recorded to be different with the current outbreak. Some of these may be associated with routes of infection. However, it may also be associated with the fact that this is the longest human-to-human transmission chain we've ever seen for this virus. And it's still the questions still remain about how that may be affecting the DNA of the virus and how it's evolving over time. Next slide, please. So just a couple of quick summary slides about this. So far globally, we've seen almost 86,000 cases. This is as of a couple of weeks ago now. The virus has been detected in 110 countries since 2022, and 103 of those countries have not historically reported Mpox. Next slide. So here in the United States, we've had over 30,000 cases be detected from all 50 states, as well as Washington, D.C. and Puerto Rico. Fortunately, we have seen a pretty serious decline in cases. The peak of infection was in July and August into September. You can see the solid line there is a 7-day moving average. And right now we're averaging around 2 cases per day, with the number of cases being drastically reduced, as well as the number of locations, obviously. This is definitely in big thanks to people at the local, state, and federal levels. And a strong effort has been made to reduce the number of cases. Next slide. So when we start talking about orthopoxvirus host systems, we don't always have information about which animals are maintaining these viruses in the environment. And that holds true for monkeypox or Mpox. However, there is significant evidence to suggest that small mammals, particularly rodents of the genus Chrysothomes and Phanecyurus, pouch rats, and squirrels, may be responsible for maintaining this virus in the wild. However, we know this virus does have a fairly broad host range and can infect a wide variety of mammalian taxa, including primates, anteaters, and elephant shrews. Next slide, please. So I guess one of the main questions we want to address here is what is the current evidence of human to animal transmission of Mpox virus, given that there are so many human cases now spread across the globe? There have only been 2 reports noted to date. The first one was from France. This was probably the big report that made the biggest splash. This paper came out back in August of 2022, reporting that a dog became infected after living with 2 men who had Mpox. Samples tested positive from lesion material, oral and anal swabs by PCR. However, the CT values were high, indicating a low viral load, but there was quite a bit of sequence data collected as well. However, the authors of this report are now noting that there is no serological response in this animal as of 26 days post-positive PCR test and beyond, and they're writing an update to their publication that should be released soon. They are now stating that they believe this is surface contamination coming from the people in the home with Mpox, rather than the animal actually being infected itself. So the other report is coming from Brazil. This is a similar situation where a dog was living in a home with people with Mpox and again tested positive by PCR. But we've heard from our colleagues in Brazil that this animal also had a high CT value, and no publication has been released yet, but we're hoping more further information will be available soon. Next slide, please. So really just to kind of talk about, when you're thinking about considerations for testing animals, this slide really sends home a big point. The samples that you use for testing animals or people for Mpox varies across the course of infection or the course of disease. So the samples that may be collected during the incubation period or when the animal or person has a fever may be different than samples that are collected when the lesions and rash is present. And so you can see sort of the first panel of this figure here is clinical markers. So the middle portion is samples for PCR, and then the third section is the serological profile. However, the serological profile is not known for many animals. This was actually developed for humans, but it does hold true for what we know about certain animal model species. So sample types for swabs include oral and nasal swabs. There's a lot of work right now to look for whether or not anal swabs are a good diagnostic marker. We do know from some laboratory challenge studies that animals can shed virus through the oral and fecal routes. And so we're trying to learn more about which sample types are best for diagnosis in animals. However, we do know that lesion material, both in humans and animals, is really the best sample type if you can collect samples during the infectious period when lesions are present on the skin of the animal or the person. Another consideration is if you get out in the period of convalescence after the animal or person has completely recovered from disease. At that point in time, no more infectious material can be collected, and you will not be able to detect viral DNA or viable infectious virus at that time. At that point, serology is really the only data you can rely on. And for animals, we do not have an IgM assay. We only have IgG assays. So we can detect IgG antibodies, but at this point in time, these serological assays are not specific for Mpox infection and can cross-react with following exposures or infections with all orthopox viruses. Next slide, please. So I want to tell you a little about a study that we're doing here at CDC with our partners at a few different states and jurisdictions. This is to basically go into homes with people with Mpox and collect samples from their animals to see if it's to try to detect any transmission of virus from people to animals. So our inclusion criteria for this study was number one, an animal had to be living in the home of a person with Mpox, and that animal had to be of mammalian origin. The other criteria was that the initial samples have to be collected within 21 days after the last contact between the animal and the person with monkeypox or with Mpox. So we had plans to collect specimens at 2 time points. First time point is the initial time point. This is collecting samples from oral and anal swabs, fur and paws, as well as some environmental samples. And we're also collecting serum at that time to get a baseline serology reading. Then we had a second sampling period that can come in 2 to 4 months after the initial sampling session. And that primarily is to only get serum for serology to look for seroconversion over time. Next slide, please. So a big thank you to all of our partners that agreed that worked with us on this project. Of course, DC Health, as well as public health departments in Virginia, Minnesota, and Tennessee. Thank you all. I'm sure some of you may be on the call right now. So we were able to sample 34 pets from 19 different households. So not a huge study sample size here, but definitely some important information that we can in this study. Next slide, please. So this is a quick summary of the laboratory results, specifically focusing on PCR at this point. So we tested many different animal samples. You can see actually we have samples from the actual animals themselves, as well as the environmental samples, which I mentioned earlier, which oftentimes came from animal bedding, food and water bowls, and toys. All of these samples came from 19 different households, as I mentioned previously. All of the numbers in the table, as well as in the figure below, These are at the individual sample level, so just comparing all different samples. And there was a significant correlation here between the samples that test positive for Mpox DNA and samples that show RNase P amplification. Now, RNase P is a human DNA marker. So in this case, we're trying to directly show that the PCR positivity for Mpox was related to the person in the home with Mpox rather than the animal actually being infected itself. And we actually see a correlation between the CT values between the Mpox assay and the RNase P assay. So further evidence that this is coming from the people and rather than from the animals actually being affected. The final piece of evidence is from the environmental samples. And actually all 5 of the animals that tested positive also tested positive with the RNase P assay. And this is added on to further by the fact that within the households, all 5 of the households that had positive animals also had positive environmental samples. Whereas out of the 14 households that did not have any positive animal samples detected, only 1 of those households had positive environmental sampling. So we're looking to analyze this further to have comparisons. We also conducted a questionnaire associated with this study to try to assess contact between people with animals in the home. So after we wrap up the study here in the next few weeks, we will begin to analyze this data further, and we'll hope to have something to share more later. I would like to say quickly, though, that we have not had any animals seroconvert through this study. We have not seen any animals develop antibodies over time, in other words, and we have not had any animals confirmed to be infected with Mpox here in the United States. Next slide. So just want to put these resources up for everybody. Of course, a big part of what we've been doing on the One Health team for the response is messaging, getting information out for people about how to protect their animals from Mpox, as well as what to do if they think their animal may have contracted Mpox. And so I think next slide, please. That's all I have today. Of course, I would like to thank everyone on the CDC One Health team, and I'll pass it back to you, Laura. Thanks.
Thank you for that presentation. Next slide, please. Our final presentation today, Salmonella species persistence on surfaces, is by Dr. Sonia Hernandez. Please begin when you're ready.
Hello, good afternoon. Can you hear me okay?
Yes.
Okay, thank you. And first, I'd like to acknowledge that the work that I'm going to talk about today was implemented by 2 students, Olivia Seiverts and Kimberly Perez, who actually did the in-field work. I also want to acknowledge Susan Sanchez, who provided us with a Salmonella isolate, and most importantly, Erin Lip, who's providing the lab support for most of the Salmonella isolations. Next slide, please. So I think most people understand that the genus Salmonella is a gram-negative enteric bacteria that has a worldwide distribution. It's one of the most common causes of diarrheal disease of both people and animals. It includes more than 2,500 distinct serotypes, most of which really don't show a lot of specificity for their host species. Specifically, Salmonella typhimurium is the serotype that causes the majority of avian salmonellosis outbreaks and can cause high mortality rates in affected songbirds and aquatic birds. Among songbirds, avian salmonellosis is widespread. It's an annual problem with certain years that lead to explosive mortality events in the United States, Canada, and in the UK. Next slide, please. There are 2 scenarios upon which Salmonella tends to cause disease in wild birds and specifically mortality events. One are young birds that are born in large nesting colonies of mixed species, typically ibises, herons, etc. But the type that really leads to more of a One Health and public health concern is mortalities that occur when songbirds aggregate at bird feeders or agricultural sites. Next slide, please. The transmission among birds has been shown to be primarily through direct feeding, as you can see in the middle, sort of horizontal transmission, or most commonly through picking it up from environmental sources at sites like landfills, dung piles, wastewater discharge areas, sewage lagoons, etc. Whereas vertical transmission is highly unlikely, or at least so far has been unproven, but certainly has not been tested on all species of birds. Next slide, please. Particularly because avian salmonellosis has such a strong seasonal component, of which we still don't really understand if there is a reservoir or if it's all associated with human behavior. Central to understanding the epidemiology of Salmonella at particularly the backyard feeder is its persistence in the environment. We know that Salmonella can persist in moist, warm conditions, especially in contaminated environments like poultry manure or bodies of water. But most of the studies that have been conducted on persistence have either been done under controlled conditions in a laboratory setting or environments that aren't really relevant to songbird health, especially as they may relate to reservoirs for public health. Next slide, please. So I and my collaborators have been studying two systems that lend themselves for understanding the epidemiology of Salmonella. For over a decade, I have been studying the trade-offs that the American white ibis, which is a medium-sized wading bird, sort of iconic to the Everglades ecosystem in South Florida, but also common along the Gulf states and up to the Carolinas. Trying to study the trade-offs that this bird faces as it adapts to living in cities. In South Florida, it's learned to adapt to living in urban environments. One of the things that we have found through our studies is that the prevalence of Salmonella shedding by ibis is much, much higher by both adults and especially by nestlings that live and utilize urban habitats. Yet the serotype and genotype diversity of Salmonella that we find in these birds is very high, indicating likely that they mirror what is in their immediate foraging environment. In one study, we found that more than half of the isolates that we collected over 2 years of sampling quarterly in South Florida matched human isolates in the PulseNet database. And one of the reasons why we consider this to be a public health concern is that white ibis are very successful in cities because people feed them directly and have a lot of contact with them in shared urban parks. Now, most avian salmonellosis outbreaks in songbirds are typically not deeply investigated. As I said before, they occur regularly and annually, and in fact, have been on the rise over the last couple of decades. We did investigate one particular outbreak back in 2009, 2010, by isolating Salmonella from birds that died. These were birds that were aggregating at bird feeders during a time that there was a concurrent peanut-associated human salmonellosis outbreak. Although the strains that we found in the birds and the peanut were not closely related at all, we did find that several of our isolates of Salmonella typhimurium matched human isolates reported to the PulseNet database. And at that time, we started to consider that feeder-associated salmonellosis was likely an underestimated public health concern. Next slide, please. And in fact, in 2021, the CDC reported a multi-state salmonellosis outbreak in people caused by Salmonella typhimurium that had a high hospitalization rate and was linked to handling sick birds or bird feeders. Thus, a better understanding of the ecological interactions between Salmonella typhimurium, avian hosts, the environment, and other animals and humans is really essential to reduce the burden of human illnesses due to Salmonella in the United States and other countries. And in fact, in Sweden, this link has already been established much better. And I'll talk about that in just a bit. But now I'm going to talk about the experiments. Next slide, please. So these two experiments are kind of small experiments, but sort of illustrating the points I made so far. The first one is one we did on white ibis. And I want to highlight that both of these experiments were conducted during relatively cold temperatures. This white ibis experiment was conducted in December in Palm Beach County, when the temperatures are for South Florida, relatively low. So to mimic what happens under normal circumstances, we encouraged ibis to feed on wooden picnic tables that were located under gazebos in urban parks. Ibis deposited fresh feces on the tables. We divided the tables up into quadrants, and then we sampled the tables every day for 8 days to determine both prevalence and persistence of each of the quadrants. And we marked some of the fecal piles as persistence piles that we followed specifically. The feces were sampled with a sterile sampling sponge that was saturated in neutral buffer, and then they were embedded in dulcityl selenite broth and then transported to UGA for processing. Next slide, please. In the second experiment, and I'm going to talk about the design of both and the methods of both, and then I'll go over the results. In the second experiment, we were actually interested as to whether feeder type, backyard bird feeder type, had any influence on the persistence of Salmonella. So first, we collected feces from chickens. We homogenized them, autoclaved them, and then inoculated them with 10⁷ Salmonella typhimurium. We had acquired an isolate that was responsible for a previous avian salmonellosis outbreak. And then that inoculated feces was transported to what we call a shade house, where typically saplings are reared, and which we converted. And then we set up 4 feeder types in triplicate, but the feeder types were randomized. The feeder types were cedar wood, plastic, and then cedar wood coated with an antimicrobial coating and plastic coated with an antimicrobial coating. Those feeder types were seeded with 1 gram of the previously inoculated feces in sterile physiologic saline. And then they were swabbed daily for 1 week, every other day for 1 week, and weekly for an additional week. Next slide, please. I think next slide. There we go. For both of the experiments, we did the same type of processing for a Salmonella isolation. We used a standard double enrichment protocol with a primary enrichment in dulcityl selenite broth, a secondary enrichment in RV broth, presumptive identification on XLT4 plates, and then confirmation of Salmonella on chrome agar plates. Next slide, please. So now just to go over the results for the IBIS experiment, we ended up sampling 96 fecal piles. The overall prevalence of Salmonella on all tables and all quadrants was 27%. I want to highlight that this is interesting because over the years, and we've been sampling adult ibises for Salmonella for, like I said, for many years now, and our prevalence is almost always anywhere between 25 and 27%. And the average persistence of Salmonella on the tables was about 3 days. 4 days was the maximum. Now, that may not seem like a lot, but I do want to highlight that after a day or so, the feces really don't look like fecal piles. They're pretty much just white and brown stains on the tables. And it was not unusual for us to see people sitting down and eating their food at these tables, even when those fecal piles were dried. Next slide, please. All right, for our feeder example, we collected 33 samples per feeder type, so you'd have to multiply that times 4 overall. And there is a mistake on your slide. The overall Salmonella prevalence was much higher on plastic feeders, and that was 12%, not 15%, and that is compared to wooden feeders, for which the prevalence was about 1%. The persistence was about 2 days, and it was 5 days maximum, again, on plastic-coated feeders and plastic feeders. Although I will say that there was a rain event on day 5 after some of the feeders had already tested negative that likely encouraged Salmonella to emerge from like a viable but yet dormant sort of biofilm state. Why do we think that the wooden feeders or that we recovered more Salmonella from the plastic feeders, which was the opposite of what we had predicted? Well, cedar wood, likely because of its composition, likely has some antimicrobial properties, and it's probably responsible for that. The antimicrobial coating that I mentioned at the beginning during the experimental design slide is a proprietary product that was produced by the engineering department at UGA, and it did not have an impact on Salmonella growth, which was also interesting. Next slide, please. So we will continue to look at other details about persistence. These were only 2 small pilot projects, but nonetheless, I wanted to offer some notes about management. We are in the middle of the season for Salmonella that typically goes from about February to May. During that time, if birds look sick or die at the feeder, it's important to stop feeding birds during that time. It is also important for people to understand that certain designs of feeder promote transmission more than others. The tube feeder that you see on this picture allows for feces to fall on the ground and not contaminate other places where birds are going to be picking up food, so they're better. And then, of course, I mentioned that cedar wood seems to be better at not promoting Salmonella growth. Obviously, also eliminating point sources of contamination would be important. And because we know from the studies that have been published from Sweden that there is a direct link between birds that are caught by outdoor cats and brought indoors and then transmitting Salmonella to people, that it is important to keep cats at all times, but especially when there might be sick birds at the feeder indoors. Next slide, please. And lastly, one of the things that we want to achieve with our communications is to make it clear to people that in order to keep themselves and the birds healthy and safe, they should practice cleaning their feeders on a regular basis. During the winter, this may need to be done almost on a weekly basis. First, the feeders need to be cleaned with warm soapy water to remove all the organic material and dispose of it. And then the feeders should be cleaned with a 10% bleach solution. And you should use implements like bottle brushes that can get to all the nooks and crannies. Then the feeders need to be rinsed and dried properly. And it is best to set feeders up in areas that get direct sunlight, because that likely has an impact on salmonella growth. And as I mentioned, to take them down during active outbreaks, and for people to handle them either with gloves or to wash their hands thoroughly after handling feeders. Next slide. And again, thank you very much to the CDC One Health team for the opportunity to get this information out there, and to the students that were involved in these studies. Thank you.
Thank you, and thanks to all of today's presenters for your informative presentations. Let's go to the next slide, please. So we do have links to resources from each presentation that are available on our website at cdc.gov/1health/zohu/2023/march.html. We do have time for a few questions. Please use the Q&A feature in Zoom to send your questions and include the presenter's name or topic. So let's start with a question for Dr. Hennessy. Have any importation regulations been considered for cats? I know that we have many for dogs, but would stronger importation regulations for cats help to prevent the spread or the introduction of sporotrichosis into the United States?
Hey, yeah, thanks so much for that question. We are indeed working with other governmental partners to try to see how we can better address some of these importation concerns. But you're right, for the moment, the importation regulations are less stringent for cats compared to dogs. But in the meantime, we're trying to work on better surveillance in order to try to rapidly detect and respond to cases in the event that they are imported, as well as our branch is working on better diagnostic testing to be able to test cats who may have suspect cases of sporotrichosis.
Thank you. And then our next question is for you, Jeff. We had a question about the change in name of monkeypox to Mpox. Do you mind speaking a little bit about the reasoning for that?
Sure, that was basically done to reduce stigma of people with Mpox infections. People felt like calling it the Mpox or calling it monkeypox was not most appropriate, and so WHO decided to go with Mpox for that. So that's the reason. Thank you.
Thanks. And a follow-up question for you as well, Jeff. Can you speak a little bit about whether there are subclinical or asymptomatic cases of Mpox?
That's a good question. I don't know that we have data for that specifically. I know that there have been cases with decreased symptoms, and many times people maybe had Mpox infections and they don't have any symptoms before the onset of the lesions or the rash. But I'm not 100%. I don't know that there have been completely asymptomatic cases, so I can't answer that question 100%. Sorry.
Thank you. Our next question is for you, Dr. Hernandez. A couple folks asked about whether the tables that were sampled were isolated so that other people or animals couldn't access them in between sampling events.
That's a great question. So in the Florida experiment, we did not prevent people from using the tables. As far as animals are concerned, the only other animals that are in the parks regularly would be other songbirds, for which we can identify the feces. Other animals that create big fecal piles like that would be ducks, and they do not get up on tables unless somebody actually physically puts them up there. But other things like squirrels, etc., don't produce feces that look the same. So in short answer, yes, we were looking to make sure that the feces look like ibis feces. We can't guarantee that that's 100% the case. In the feeder experiment, we wanted an enclosed environment, but one that still had access to sunlight and temperature, etc. And that experiment was conducted in February, if I didn't mention it before. And no, that house was completely sealed off. In fact, we added mesh, etc., so that it could not be accessed by songbirds or lizards or anything of the sort. But thank you for the question.
Thank you. We do have another question for you. Have you done any research or looked at any other pathogens in addition to Salmonella, like Campylobacter, perhaps?
I have not looked at Campylobacter so far. It is one that we've talked about quite a bit. The study with the ibis is a large ecological study that looks at a variety of different pathogens. We did look at Vibrio. There are some interesting findings there, but I can't share them here now. But Campy would be yet another one to look at for ibis. I don't think there are that Campy is a big concern for songbirds.
Thank you. And we have another question for Dr. Hennessy. Have you found any correlations between whether a cat is kept inside, outside, or is an indoor-outdoor cat in terms of the potential spread of sporotrichosis to people?
Yeah, thank you for the question. I think in terms of just risk for contracting the disease, generally, at least in Brazil, we mostly see S. brasiliensis in cats that have access to the outdoors, be that stray cats or owned cats that are indoor, outdoor, or mostly spend their time outdoors. And I think that risk would then correlate to humans. If you have an indoor cat that doesn't have any risk of getting the disease, then you as a cat owner would be less at risk. I'll say there is the one exception. Occasionally, like in this case from the United Kingdom, the cat was brought from Brazil 3 years previously and was otherwise healthy until like a few months prior to these humans getting it last year. So it was an indoor cat, but it had previous outdoor exposure in Brazil. So in that case, it then presented a risk to its owners. But I think generally outdoor cats are more at risk for getting it and then maybe transmitting it as well.
Thank you. And then we'll go back to Dr. Hernandez for our last few questions. Are there any recommendations around wastewater treatment plants due to birds wading, in wastewater ponds, things like that?
Yeah, I can't say 100% because I don't know this, that wastewater treatment plants in their first stages, right, where you have the most contaminated, are doing something to disperse wildlife. I will say that constructed wetlands have forever been touted as prime wildlife habitat, particularly since natural wetlands are in decline in so many places, and constructed wetlands are a big source of habitat aggregation in Florida, where I spend quite a bit of my time. And there have been some studies to look at, for example, antimicrobial resistance determinants and a few other enteric pathogens from those type of systems. So I think it's worth balancing the trade-offs between the habitat availability and the seriousness of a source for contamination.
Thank you. And then I have a follow-up question for you as well. Could you talk a little bit about the risk? You talked about the public health risk to people from Salmonella, but what about the risk to pets, dogs and cats? Is there any risk of a dog or a cat getting sick after being in contact with birds around bird feeders?
Yes, and I'm sorry if I did a poor job of describing that, but there are actually several publications in which especially cats have gotten infected, some of them asymptomatically, and some which the direct line between the bird, the cat, and the person has been beautifully outlined. The most recent one, I think, in 2021 from Sweden, where they did a really nice investigation following that. So, and the problem with birds that get salmonellosis is that they can be sick and sort of, fluffed down and sitting on the ground for a couple of days, which would make them very vulnerable to any domestic animal to come along and snatch them up.
Great, thank you. If you have other questions for today's presenters, we've included their email addresses on this slide. They're also on the ZOHU call webpage for today's webinar and in today's ZOHU email newsletter. And as a reminder, a video of today's webinar will be posted within 30 days. Next slide, please. Thank you again to all of our presenters and our participants. We hope you join us for the next ZOHU call on April 5th. Thank you for your participation. This ends today's webinar.